In this video, we are gonna see about major histocompatibility complex by Dr. Gayathri. The MHC molecules are glycoproteins encoded in a large cluster of genes located on chromosome 6. They were first identified by their potent effect on the immune response to transplanted tissue. For that reason, TH gene complex was termed a major histocompatibility complex. MHC genes called the H2 complex in mice were first recognized in 1937 as a barrier to transplantation in mice. In humans, these genes are often called human leukocyte antigens LA, as they were first discovered through antigenic differences between white blood cells from different individuals. MHC is the term for the region located on the short arm of chromosome 6p1, 31 in humans and chromosome 17 in mice. In humans, it contains more than 200 genes. The human leukocyte antigen HLA region, also known as the major histocompatibility complex MHC on the short arm of chromosome 6. The HLA is crucial to immune recognition of self and non-self peptides, a complex of genes encoding cell surface molecules that are required for antigen presentation to T cells. There are three classes of MHC molecules: Class 1, Class 2, Class 3. Fundamentally important: V basis of self, non-self distinction, Y presentation of processed antigen, MHCI on nearly all nucleated cells, MHC on B cells, macrophages, dendritic cells. Class 1 and I are distinct structural entities, although there are multiple Class 1 and 2 genes. All the gene products have similar overall structure. Class 2 is a diverse collection of more than 20 genes, including those encoding complement proteins. The HLA contains lots of genes close together that are transferred from the parent to the child in adjacent haplotypes that are said to be in linkage, disequilibrium. MHCI, two chains, heavy chain alpha and small chain microglobulin beta 2 heavy is 45 kilodaltons, has three extracellular domains called A1, A2, and a 3CI and X to interact to present process AG monomer, a transmembrane component 40A, a cytoplasmic tail 3OAB2, microglobulin 12KD associates non-covalently with A3. Part of immunoglobulin superfamily is said to microglobulin is the only member of the superfamily that does not have a component linking it to a membrane. MHC, one alpha and beta chain, 33KD and 28KDA, respectively. Non-covalently associated, each chain has two domains. AR interact to present processed AG13 to 18AA. O2 and B2 are part of immunoglobulin superfamily. The major differences between the two MHC class molecules lie at the ends of their peptide binding _clefts, which are more open in MHC molecules compared with MHCI molecules. The MHC molecule cleft is made up of a non-covalent association between the AR and BR domains, and that binds the peptide through multiple van der Waals forces and hydrogen bond. The main consequence of this difference is that the ends of a peptide bound to an MHCI molecule are buried within the molecule, whereas the ends of peptides bound to MHC2 molecules are not. This difference allows more flexibility in the length and types of peptides that MHC molecules can bind. Peptides that bind a specific class to molecule will share the same middle anchor residues but may vary in length and sequence of other residues. There are no rearrangements. MHC proteins from the haplotype constitute a lifelong cell surface character for any vertebrate. This circumstance is very different from X, which are constantly being generated in response to new foreign proteins and carbohydrates in the environment. The loci which specify MHCs are polymorphic. Many alleles may exist at a locus. HLA a locus, approximately 60 alleles. HLA-B locus minus 110 alleles. HLA-C locus, approximately 40 alleles. The high level of allelism creates diversity within a species, thus restricting allografting, but does not produce diversity within an individual. Why polymorphism and polygenism in MHC? Polymorphism and polygenism in MHC make it impossible for a pathogen to alter its antigenic epitopes so that the epitopes cannot be presented by a majority of the individuals in a population. Most individuals have 6 MHC class I proteins and at least 6 MHC class IL, so if one MHC protein cannot present a pathogen's antigens, another MHC protein probably can. Each species has thousands of ways to present antigens from a pathogen, so even if some individuals or small populations cannot efficiently present some antigens, the species is not at risk. Functions of MHC molecules. Asterisk antigen processing is the modification of native proteins into peptides for presentation by MHC. MHC class I presents processed proteins that were in the cytosol. Antigens are recognized by CD8 T cells, TC cells CTL. MHC class II presents processed proteins that were in endocytic vesicles. Antigens are recognized by CD4 T cells TH.